Hello and welcome to Insight Ophthalmology. This is Dr. Amrit, welcoming you to another lecture. Today we are studying about the vitreous embryology and its disorders. The vitreous cavity is the largest cavity of the eye and it actually forms about 2/3 of the volume of the eye. It is situated behind the lens and the ciliary body anteriorly and the retina posteriorly. Its weight is about 4 grams and the volume of the vitreous cavity is about 4 cc. In the video on development of eye, we discuss in detail about the optic vesicle and the development of the lens. The link is going to be provided in the description box. In that video, we discuss how the neural tube and specifically the anterior part of the neural tube give rise to outpouchings which are going to develop into the future optic cup. Now, before the optic cup develops, we have the optic vesicle and the optic stalk. Now, eventually what happens is that a vessel starts developing ventral to the optic stalk and the optic vesicle. And this vessel will basically go inside and supply the developing lens, and this developing lens is known as the lens vesicle. And the vessel which is developed here is known as the hyaloid vessels. Now, in order to give way to the hyaloid vessels into the optic cup and the optic stalk will start developing a groove inferiorly and they will ultimately develop this fissure over here. And this fissure is known as the choroidal fissure or the optic fissure. The purpose of the choroidal fissure or the optic fissure is basically to allow the hyaloid vessels to grow inside the optic cup and reach the developing lens vesicle. Okay, so that is very important if you want to understand about the development of the vitreous. The following slide actually tells you about the same thing. So we have the optic stalk, the optic cup, and you can see this fissure on the ventral aspect known as the choroidal fissure. And the choroidal fissure basically allows the hyaloid artery and the hyaloid veins to enter into the optic vesicle and ultimately reach the lens vesicle and supply the lens. Okay, the reason this is important is because we should know that embryologically there are basically 3 types of vitreous. We have a primary vitreous, we have a secondary vitreous, and we have a tertiary vitreous. Now, the primary vitreous is formed first, the primary vitreous is then replaced by the secondary vitreous, and then we have the tertiary vitreous which is formed out of the peripheral area of the secondary vitreous. Now, the first type of vitreous that we are going to discuss about is the primary vitreous. The hyaloid artery which has basically entered through the choroidal fissure and which has basically reached the optic vesicle and ultimately the lens vesicle is responsible for the formation of the primary vitreous. As a matter of fact, it would be not wrong to say that the primary vitreous is predominantly formed by the hyaloid artery and its branches. The hyaloid artery branches are also known as the vasa hyaloidea propria or the vascular mesenchyme. Okay, so at about 5th week of gestation, the space between the lens and the retina gets occupied by this primary vascular vitreous, which is basically formed by the hyaloid artery and its branches. So as the hyaloid artery is basically mesenchymal in origin, and since it's coming into the choroidal fissure and reaching the lens and ultimately forming the primary vitreous, it would not be wrong to say that the primary vitreous is partially mesenchymal in origin. It is partially mesenchymal because it also has some ectodermal elements. From the first diagram, you can see that here we have the surface ectoderm, and the surface ectoderm thickens at a location where the optic vesicle approaches it, and that forms the lens placard, and ultimately the lens placard is going to form this lens pit and ultimately the lens vesicle. Now, as the lens vesicle gets pinched off from the surface ectoderm, some of the cells of the surface ectoderm will also get pinched off and they will move behind the lens, and ultimately they will lead to the formation of the primary vitreous. And therefore, we can say that the primary vitreous actually has a mixed origin. It is partly ectodermal in origin and partly mesenchymal in origin. So I hope that is clear. But remember that primary vitreous is basically the vascular vitreous. Now, as we discuss about the primary vitreous, which is vascular vitreous, it is important to discuss also about the tunica vasculosa lentis, that is the plexus of the vessels which are going to form around our lens to supply the lens during the embryological period. The tunica vasculosa lentis starts developing at about 5th week of gestation. And there are 2 arteries which are feeding the tunica vasculosa lentis. We have the posterior ciliary artery, supplying basically the anterior part of the lens up to the equator, and the branches of the posterior ciliary artery will form what is known as the lamina iridopupillaris or the pupillary membrane. It is called the lamina iridopupillaris because it is in close association with the iris and the pupil. Now, the posterior component of the tunica vasculosa lentis, that is beyond the equator and behind the lens, is formed from the hyaloid vessels, and they are going to form the capsulopupillary vessels, okay, because they're supplying the posterior part of the capsule at the equator and posterior to it, right? So that is the tunica vasculosa lentis. So the beginning of the primary vitreous is at about 5th week or 35 weeks of gestation, where you can see how the hyaloid vessel is actually entering and forming these branches known as the vasa hyaloida propria and supplying the posterior part of the lens up to the equator, and the anterior part of the lens is basically being supplied by the long posterior ciliary arteries which are coming like this, and together they are forming the tunica vasculosa lentis in the anterior part of the eye, and this hyaloid artery which is present behind is going to take care of the posterior part of the vitreous. Now together this is known as the primary vitreous, right? So primary vitreous is predominantly formed from your hyaloid artery. However, not long that this primary vitreous is going to be now resorbed by the appearance of specialized cells, and these cells are known as the hyocytes, the cells of the vitreous. Right now, these hyocytes are also mesenchymal in origin, and these hyocytes actually have phagocytic properties. That means they can eat up the primary vitreous, and they start eating up your hyaloid arterial system. And now, as they eat up the hyaloid arterial system, they also start depositing the collagen fibrillary material within the vitreous, and now the vitreous that is formed is known as the definitive vitreous or the avascular vitreous or the secondary vitreous. And the time period during which it is formed is between 5.5 weeks to 12 weeks of gestation. Okay, so I hope that is clear. So that is your secondary vitreous. Now, you should know that the adult vitreous basically is about 99% water. Apart from that, we have collagen, we have other soluble proteins, and we have basically the extracellular matrix formed of the glycosaminoglycans and hyaluronic acid. Now, all these constituents are most of the hyaluronic acid and the collagen type 2 in the vitreous gel is added after birth, although most of the vitreous, the secondary vitreous or the definitive vitreous, is formed during the embryological period. Most of the hyaluronic acid and the collagen type 2, which forms the major type of collagen in the vitreous, will be formed after birth. So I hope that is clear. Now that we know what is meant by the secondary vitreous, you should know what is meant by tertiary vitreous as well. Now, tertiary is the third vitreous, so you can roughly remember that it comes at around the 3rd month of gestation. Now, basically what starts happening is that the secondary vitreous starts condensing in the peripheral portion, specifically in the area of the pars plana. Now, from the anatomy of the ciliary body, you would remember that we have the iris, we have ciliary body. We have two parts of ciliary body. The anterior part is called the pars plicator, and we have the posterior smooth part called the pars plana. Now, the tertiary vitreous basically starts developing these firm attachments with the epithelium of the pars plana, and ultimately in adulthood or when it gets matured, it is going to form what is known as the vitreous base. Right? So tertiary vitreous ultimately leads to formation of the vitreous base. Okay. Now, we discuss about the origin of the primary vitreous, which is a mixed origin. We discuss about the laying down of the secondary vitreous, which is coming from the hyocytes, right? And now we should also know what is forming the tertiary vitreous. Now, it is believed that the non-pigmented ciliary epithelial cells in the region of the pars plana are actually responsible for the formation of the tertiary vitreous, and they also give rise to the zonular fibers, right? So knowing about the origin of the vitreous is very important. You should know where is the primary vitreous coming from. You should know where is the secondary vitreous coming from. And you should also know the source of the tertiary vitreous. So I hope that is clear. Having discussed about the embryology of the vitreous, now let's discuss about the abnormal development of the vitreous. Okay, so first let us talk about the disorders which are going to develop because of the abnormal atrophy of the primitive structures. Now, all the primitive structures are supposed to involute. So, what I mean to say is, initially we have the primitive vitreous, which is the primary vitreous, that has to be replaced basically by what? It has to be replaced by the secondary vitreous known as the definitive vitreous, right? So, during the fourth month, whatever remains of the primary vitreous are there, they also begin to atrophy. Okay, the vessels basically which are coming from behind and reaching the lens, basically the course of these vessels through the vitreous will be evident in the adult life as a very narrow central fluid filled canal. So that narrow central canal is known as the Clockett's canal. So the path that was taken by the hyaloid vessel to reach the lens will be now replaced by a small canal without any hyaloid vessel in it, and that is known as the Clockett's canal or the hyaloid's canal. Okay, now basically it is the macrophages which play a very important role in the regression of the primary vitreous. I already discussed about that. These macrophages are nothing but your hyelocytes, right? So that central vitreous channel that you see over here is known as the Clockett's canal, also known as the retrolental canal. It extends from the posterior surface of the lens to the optic nerve head, and it represents the remnants of the lumen of the course of hyaloid artery during the embryogenesis. Now, it's very important to know that this space is devoid of collagen fibrils, and it is about 1 to 2 millimeters in diameter and has an S shape from the anterior to the posterior end. Now, as you can see over here, the Clockett canal basically starts from the patella fossa. Now, if you would know, if you would have seen our video on the anatomy of lens, you would know what is meant by patella fossa. So, patella fossa is nothing but it is just an anterior depression in the vitreous where the lens basically sits, right? So, this depression anteriorly is known as the patella fossa. So, our Clockett canal basically starts from the patella fossa as a very slight widening like this, and then it takes its S-shaped course in the central vitreous, and again it widens near the optic nerve head, and this widening or the funnel shaped widening is known as the space of Martigiani. Okay, so I hope you understood what is meant by space of Martigiani. Next, what we are going to talk about is the malformation of the vitreous and the hyaloid artery system. So, it's very important to revise the primitive vitreous anatomy or the primary vitreous anatomy. We know that it is majorly vascular, and we have these hyaloid artery and the long posterior ciliary artery which are actually going and supplying the lens. Now, if you would carefully observe, you can arbitrarily divide it into an anterior portion and a posterior portion. The posterior portion is majorly formed by the hyaloid artery and its branches, whereas the anterior portion is actually being supplied by the long posterior ciliary arteries. Now, the long posterior ciliary arteries are actually forming the pupillary membrane, whereas the hyaloid artery is supplying the posterior aspect of the vitreous here and the posterior aspect of the tunica vascularis lentis up to the equator, and at the equator they are basically forming the capsulopupillary membrane. Okay, now together the pupillary membrane and the capsulopupillary membrane is known as the tunica vasculosa lentis. Okay, now normally, the hyaloid system of vessel will vanish completely. However, in some disorders, the regression does not occur and these vessels will persist, causing some problems. Now, what happens is that whenever the vessels are persisting, usually some amount of collagenous tissue will also persist along with them, and such fibrovascular tissue are later going to cause contraction on the surrounding structures and traction. They are going to contract and cause traction on the surrounding structure, leading to deformation of the normal tissues of the anterior and the posterior segment. Now, if your anterior part or the vessels on the anterior part are basically persisting near the lens, it is known as the persistent tunica vasculosa lentis. And if the hyaloid artery in the posterior aspect of the eye is persisting, that is known as the persistent hyperplastic primary vitreous or the PHPV. Okay, now let us see a few examples. So here what you can see is the persistent pupillary membrane. So here just the anterior part of the tunica vasculosa lentis is actually persisting. Here what we can see is fine iris strands which are actually bridging the pupil, and they are also connected or they are also attached to the lens. Now in most of the cases, the persistent pupillary membrane will arise or will be attached at the collarette of the iris. Okay. So by this time, I assume that all of you know what is meant by collarette, and if not, you can actually visit our video on the anatomy of the iris. Okay, now the persistent pupillary membrane, they are usually asymptomatic um, and they do not cause any functional disorders in the individuals who have this persistent pupillary membrane. However, in rare cases, there might be really dense membrane, and these dense membranes can persist and they can obscure the pupil. And when they obscure the pupil, they can actually cause what is known as amblyopia. Okay. However, in some portion of the pupillary membrane might be seen in normal individuals as well. However, in those people, it is usually asymptomatic and does not cause any functional problem. Now, if the posterior aspect of the hyaloid system actually persists, that is known as the persistent hyperplastic primary vitreous. And here again we have two varieties, the anterior persistent hyperplastic primary vitreous or the anterior PHPV, and we have the posterior PHPV. Now, in the anterior PHPV, basically the part of the hyaloid which is supplying the posterior aspect of the lens up to the equator persists, whereas in the posterior PHPV, it is the posterior part of the hyaloid artery that basically persists. So here you can see that this is a diagram depicting the anterior PHPV. You can see that here we have this persistent anterior fibrovascular membrane because of the persistence of the anterior part of the hyaloid artery, which is forming the posterior part of the tunica vasculosa lentis. So this is known as the persistent anterior primary vitreous. You can see the posterior part of the stalk is basically thinned out, whereas the anterior part of the hyaloid is persisting, right? So obviously here you will see the anterior fibrovascular tissue in the anterior vitreous phase or behind the lens. So you can see over here the lens is normal, and behind the lens you see that whitish membrane, and that whitish membrane also shows certain vessels in it, so telling you that this is actually a fibrovascular membrane, and actually it is a persisting anterior fibrovascular tissue from the posterior tunica vasculosa lentis. Right now, what happens is that, as I told you, that wherever there is a vascular component, there's also a fibrous component, and that fibrous component goes and attaches to the surrounding ciliary body processes, right? So as they attach to the ciliary processes, they will also draw those ciliary process internally. So basically what I mean to say is that this fibrovascular tissue will connect or will attach to the ciliary processes, and as the fibrovascular tissue is going to contract, it's going to pull inwards the ciliary processes, making them visible through a dilated pupil examination. So normally through a dilated pupillary examination, we usually do not see the ciliary processes. However, in a case of anterior PHPV, because of that fibrovascular tissue sitting behind the lens, that is going to cause traction on the ciliary processes, making them quite prominent. Right, lens obviously will be opaque in the anterior PHPV, and this happens because this fibrovascular mass can actually erode the posterior lens capsule and also penetrate the lens cortex, right? And obviously there will be appearance of a whitish pupil, so this is known as leukocoria, and anterior PHPV is a very common differential of leukocoria, and sometimes it can be mistaken for retinoblastoma as well. Now, what about posterior PHPV? In this diagram, you can see that the lens is pretty clear. There is no, just the stalk is connected here, but there is no fibrovascular proliferation behind the lens. And here you can see that the posterior part of the hyaloid artery is actually persisting. And you can see here the retina is basically wrapping around that stalk of the persistent hyaloid artery, right? And this is known as the posterior PHPV. So persistence of the posterior part of the hyaluronic artery is known as the posterior PHPV. Now usually you are going to see this vascular structure or fibrovascular structure projecting from the optic disc to the adjacent retina. Okay, and this will also cause distortion on the disc by the prepapillary and preretinal fibrous membrane. So what did I tell you? Wherever there is a vessel which is wherever there's a vessel which is anomalous, there will also be a fibrous component associated with that vessel, and there will be contraction, and because of that contraction of the fibrovascular structure, there will be traction on the adjacent structures which are present there, and in this case it is the disc, and therefore the disc will be distorted in case of posterior PHPV. Apart from that, the retina also might be dysplastic, and there will be some radial forms, folds or falciform folds which can be seen on the retina. So falciform folds are nothing but they are the sickle shaped folds that you can see on the retina. Now here again is a picture of the PHPV. Now, PHPV is also known as the persistent fetal vasculature, and you can see over here the disc is also not normal. So you can see this band which is actually extending anteriorly. This band can actually extend right up to the posterior aspect of the lens, and the disc over here is dysmorphic. So this is an example of posterior PHPV, right? So we already discussed what is meant by the space of Martigiani. So space of Martigiani is nothing. It is a funnel-shaped dilatation of the Clockett's canal, basically near the optic disc. So that is important. And why we discuss about the space of Martigiani is that because sometimes you can find just a flake of glial tissue projecting from the optic disc. Now this is also a remnant of the hyalid artery system. It's just that you will just find that glial tissue sitting over there and not the vessel, and this is quite smaller compared to the posterior PHPV, right? So this glial tissue sitting at the optic disc is known as the Bergmeister papillae. Okay, so this Bergmeister papillae is situated in the space of Martigiani and is a flake of glial tissue projecting from the optic disc, as you can see over here. Similarly, you might find a very small remnant or at the anterior end of the hyaloid artery attached to the posterior lens capsule, and that is known as the Middendorf dot. Okay, now this dot might actually be responsible for the development of the posterior polar cataract. So that is one clinical point associated with it, right? So in this video, we talked about the embryology and the development of the primary, secondary, and the tertiary vitreous with special focus to their origins, because that forms a very important high yield question. Apart from that, we also discussed about the various disorders, developmental disorders of the vitreous that can arise because of the persistence of the hyaloid arterial system. So we discussed about the persistent pupillary membrane. We discussed about the persistent fetal vasculature, also known as the persistent hyperplastic primary vitreous or PHPV. And we ended by discussing what is meant by Bergmeister papillae and the Middendorf dot. Okay, so that's all for today. I hope that was useful. Thank you and have a nice day.